Imagine you are treating a patient with advanced stage glioblastoma. The brain tumor is surgically removed and the patient is given a regimen of chemotherapy, but still he has rapid progression of his cancer. The tumor has become too widespread for more surgery or radiation to be helpful, and it is so heterogeneous that no therapy exists to target it. We need a new approach, and systems biology may get us there. But why systems biology? And more specifically, why should we use systems approaches in cancer research? To answer these questions, we first need to talk about three things. First, we'll define systems biology. Second, we will talk about how cancer is considered a complex system. And third, we will investigate how systems approaches are used in cancer research. Together, these tell a story about why systems approaches are useful to cancer research and have the potential to be useful in the clinic. So what is systems biology?
To me, systems biology in general is asking a question or defining a problem that at the onset, by definition, is beyond one's intuition.
You can't just intuit the answer. You really need the benefit of computation, math, in order to dissect the parts and understand how they work together.
Systems thinking generates non-intuitive solutions because complex systems are more than the sum of their parts. In cancer in particular, many interacting parts can lead to unexpected outcomes, and we need systems biology to predict such outcomes.
Systems biology, to my mind, involves sort of modeling the complexity in general, but when we're talking about cancer, sort of modeling in particular the signaling complexity within a given cancer.
So systems biology is definitely, for me, the idea of trying to understand a biological system, whether it's an individual cell, whether it's how cells come together, whether it's how cells come together into organs, how organs come together into people, or how people come together into larger networks, and trying to essentially measure information about all of those pieces and then use approaches to learn about what all of those pieces mean emergently, or something that you couldn't have understood by measuring one of those things in the absence of the other things at the same time.
Systems biology is the marriage of computational models with sophisticated experimental data as part of that iterative process of predicting and testing.
Altogether, we might say that systems thinking is considering that the whole is more than the sum of its parts, and systems biology is using this thinking to evaluate non-intuitive behaviors of complex biological systems at multiple scales with computational modeling and experiments. In all of these definitions, one idea has come up a lot that explains why cancer is so hard to treat: complexity. So you might be wondering at this point, what makes something a complex system? Systems thinking is particularly useful when dealing with complexity. Modeling cancer as a complex system can help save lives. Cancer is a complex system because a relatively simple event, a mutation, leads to individual components, the mutated cells, that can interact in non-intuitive ways. These interactions give rise to unexpected emergent behaviors such as metastasis, heterogeneous phenotypes, and therapy resistance. Therapies today often don't help everyone, or they lead to relapse because they treat cancer like a complicated problem rather than a complex one. Modeling complexity at every scale can help us predict when certain patterns occur and how to prevent them, leading to lives saved. So cancer is a complex system, and those could usually benefit from systems approaches. How is systems thinking applied in practice? Let's consider a specific problem with cancer, the fact that it is constantly evolving.
One of the reasons cancer is so incredibly hard to treat is that there's essentially evolution happening in the cancer.
This evolution to a treatment-resistant disease may counterintuitively be promoted by aggressive therapies. Reducing the intensity of the therapy could also reduce the pressure for cancer cells to adapt.
There might be smart ways revealed by computation that you can schedule drugs to achieve the desired effect clinically for a patient, but to minimize toxicity.
There's the potential for systems biology to change the conversation away from the decades-long view of eradicating the disease, cutting out the cancer and killing everything else that wasn't removed. I think there's a potential to incorporate ideas of feedback, adaptation, competition in the way that we deploy therapies to patients.
One systems biologist who has been working to change the conversation is Robert Gatenby, who has developed an approach to cancer treatment called adaptive therapy.
Adaptive therapy is meant to increase the overall survival using existing drugs and lower doses to improve the quality of life.
To maximize the time it takes for cancer to evolve resistance to treatment, Gatenby says you need to consider evolutionary principles.
In this way for 50 years, standard therapy is maximum tolerated dose continuously until progression. The problem is evolutionarily, that's usually unwise. And the reason is that by applying the same treatment over and over again at maximum tolerated dose, what you're doing is maximally applying evolutionary pressures for resistance, and you're also removing their competitors.
The key insight with adaptive therapy is that the proliferative drug-sensitive populations can prevent smaller resistant populations from growing. Killing the sensitive cells in normal chemotherapy therefore selects for resistant cells, and they can proliferate because their competitors, the sensitive cells, have been killed. This is called competitive release, which is avoided in adaptive therapy.
A typical adaptive therapy approach would be to say, instead of giving maximum tolerated dose just continuously, to give a reasonable dose of therapy, just enough to knock the tumor down. And in this case, you explicitly leave behind a significant population of sensitive cells. The sensitive cells are your buddies. You can control them, and you want to use them to control the tumor cells you can't control. You can then stop therapy, and the tumor will regrow. But you'll notice that as it's regrowing, you're not putting any selection pressure for resistance.
So the sensitive cells regrow to form the same mix of resistant and sensitive cells as before treatment. The tumor is then controlled by another round of treatment.
The amount of therapy depends on what's happened to the tumor. If the tumor has grown a little bit, you give higher doses. If it's gotten smaller, you give lower doses. And you simply just keep going through this iteration. What was particularly interesting in the preclinical studies is that what we found was over time, we could maintain control of the tumor using lower and lower doses of drug to the point where we could almost not treat at all.
This approach is making its way into the clinic today, with the first clinical trial focused on treating castrate-resistant prostate cancer with abiraterone.
And that's been published, and the results were really quite good. Overall survival was increased by almost 2.5 years. Now we're going to be using androgen deprivation therapy with abiraterone, and we're going to be using this with mathematical models where the dosing is changed at every treatment based on model design. So that would really be the most sophisticated mathematically and evolutionarily trial that we've tried so far.
If you look closely, you might notice a loop between computation and real life that we touched on before. This is a hallmark of the systems approach. Data from the patient is used as input to a computational model, which can make predictions about cancer progression. In the lab, these computational predictions are then tested with experiments, generating knowledge, validating the model, and providing more data. When a systems approach translates to patient care, these same models can be used to predict the behavior of a cancer and influence treatment decisions. The patient can then be monitored, generating new data for computation. The iteration between computation and patient data can help tackle problems like therapy resistance that arise in cancer treatments today.
When we move away from thinking of a one-size-fits-all kind of approach to treating disease, I think that can be powerful.
It provides for me hope that there are ways to unlock extreme sensitivity to targeted therapy in combination with chemotherapy. And I think systems biology is probably the way to get there.
Now there's one last question we would like to answer. How can you get involved in systems biology?
Even amongst the clinician scientists like me, there are very few of us who are doing systems biology work ourselves. So really, I think the best way is to connect and collaborate with systems biologists on the research side and really work to apply those techniques to what we're doing in the clinic.
No one can know everything. And the oncologists can work with mathematicians or, with bioinformaticians and that sort of thing. And it's a beautiful thing if it works, because you learn from each other, you respect each other, and you contribute to each in its own way.
There's a lot of individuals that the first thing that they say when they come and talk with me is that I'm not a systems biologist. And my reply these days now is to say, not a systems biologist yet. Anybody who is willing to learn new things and ask questions that maybe they fear are stupid, but almost certainly are not, has the potential to grow or begin incorporating systems biology elements, systems biology thinking into their own work.
Remember that patient with advanced glioblastoma who had no treatment options? Now let's imagine a world in which oncologists use systems approaches to diagnose and treat patients. After surgical resection, you sequence the tumor and determine which therapy regimen would best fit the patient's genetically complex cancer. During the course of treatment, you measure biomarkers and use computational models to adjust the treatment plan as necessary to avoid relapse. The continuous feedback between modeling and patient data gives your patient the best chance of survival, and the glioblastoma responds to this evolving regimen. The patient's life expectancy climbs dramatically, and the use of adaptive therapy means the patient doesn't relapse. Systems biology plays an essential role in making this dream a reality.